Hi, this is a video for my Year 1 genetics class. Today we're covering chi-squared, which is quite a tough topic. It's a statistical test which is where we look at observed and expected numbers. So is what you can see what you would expect? So it could be used with plants. So is what you see what you would expect in two different areas? Are they the same? And therefore you can usually accept the null hypothesis that there is no difference. Or does it statistically show you that there is a difference between the two populations and therefore assume you can accept your experimental hypothesis? The other one that it's used for, which we're going to do today, is it's used for genetics, because we've got an expected inheritance pattern, knowing particularly Mendelian genetics, which we're doing now. And then we can look at observed numbers of offspring to see if they match the expected. Okay, so we're doing it on Drosophila, so fruit flies, not real Drosophila. I've not got real flies flying around. The two genes I'm going to give you are for wing length and for eye colour. Okay, so we're going to use a W for wing length. So normal wing length is the capital, that's the dominant, and the small W is the short wing length, which is the small w. The R's, so you've got a capital R for red eyes, and you've got a small r for white eyes, which is the recessive allele. So you would need two copies of that to get the white eyes. Okay, so hopefully you've written those. If you're following on the sheet with me, hopefully you've filled those in the boxes. If you're just listening to learn about chi-squared, then that's fine. I'll just keep referring to the sheet for my Year 1 genetics class. So if we have two parents, which we are going to cross, we've got a homozygous dominant parent and a homozygous recessive. So we've got two true breeding parents. So homozygous dominant, hopefully you're following with me and would be happy that a homozygous dominant would look like that. Therefore, a homozygous recessive, change the W just so it's C. W is one of those tough ones where you can't see if it's a capital or a small. So a homozygous recessive would look like that. So that would be the genotype. And remember, it's a being, an organism, a fly in this case. So it has 4 letters. It has 2 copies of each gene. Okay, so when it makes gametes, if you've seen my other video, you'll know that this is generally how I demonstrate. Now you'll notice in both of these cases, the results are the same. Okay, so this dad can only produce a W and an R. Okay, those two will always go together because that's all he's got. And mum has only got those 2. Okay, so we could do it 4 times, twice, to make our 4 Punnett square, but we don't need to because we know the offspring are all going to be heterozygous. Okay, so if we look at the phenotype of these, because it has got the capital W and it has got the capital R, this offspring, this first generation, will have normal wing length and red eyes. Okay, good. So then we're going to allow 2 of those to breed like we always do. So 2 of these heterozygotes. Now if you do that one for me, and again you're looking at the gametes that can be produced. So I always do that, always helps. So if you were to do your 4x4 square, remember you need a 16 square Punnett square now. I'll help you with the gametes and then we'll move on. So the gametes that each parent can produce would be capital capital, capital small, small capital, and small small. We usually put the double recessive at the end because when you do your Punnett square, because this would be the same down the side. Yeah, be the same down the side. And when you cross them and then look at the phenotypic ratios, you should get your magic Mendelian number, your Mendelian ratio of 9331. Okay, so on the back of the sheet, if you're following with the sheet, so on the second page, you should have your 4x4 square. These should be your gametes across the top and the same down the side. Do the cross in the middle with genotypes and phenotypes, and you should be able to show me with colours or some other way of annotating that you've got a 9:3:1 ratio. Okay, so that's the first half done. For the next bit, what we need to do is actually look at a population. So I've got a population of fruit flies here. Okay, so we're on page 3 now, if you're following me with the sheet. And what it asks you to do is have your representative sample, which I've got here. Okay, so instead of having flies, I've got cutout flies. It's a lot nicer in the classroom to have these. And what we need to do is separate them into phenotypes and then count how many we've got. Okay, so if you look closely here, this one is a red eye and normal wing. So if we go back up, red eye and normal wing has to have one capital R at least and one capital W. Okay. The next ones I've got, if I move all those to one side, I've got ones here. These ones have got normal wing length, but they've got white eyes. Okay, so this one would be capital W, but 2 small r's. Okay. Next one. This one here is in the population. They're quite fiddly when they get small. This one has red eyes, but small wings. So here we've got 2 small W's and at least 1 capital R. Doesn't matter what the other R is. Okay, to have red eyes. Now this one, here's the homozygous recessive. So you can see it has shorter wings and it has the white eyes. So it must have 2 small W's and 2 small R's. So this is the one where we get 1. This is our 1. Okay, so here's my population. I've split them into phenotypes. I've got 2 of those. I've got 1, 2, 3, 4 of those. I've got 1, 2, thought I had 3. Yes, I do. I've got 3 of the long wing length white eyes. And I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, I think. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yes, I have. Okay, so I've got 11, 3, 4 and 2. Okay, so I'm just going to write that below so you can see. So I've got 11, 3, 4, 2. It doesn't matter which way round these are really until we get to, because we're doing, it's a 3:3 ratio normally for the 9:3:3:1. So it doesn't wholly matter which way round they are. Okay, so what we need to find out now is this ratio drastically different from Mendel's 9:3:3:1, i.e. does it follow Mendelian genetics, or perhaps Mendel was wrong, or we've got a very strange population, or does it follow he was right, we can all rest easy that inheritance does work in this way. Okay, so out of 16, because we've got 16 babies normally for a 4x4 Punnett square, out of 16, we would expect there to be 9 of these big ones. Now, I haven't got 16 in my population, which is the next bit on your sheet. I haven't got 16, I've got 20. Okay, so to work out how many I would expect in a 9:3:3:1 ratio out of 20 flies. Okay, so how many would I expect? So you can do it various ways. The way my brain works is to do it like this. So I have 20 flies instead of 16. So I'm looking for 9/16 of 20, basically. And I know it's going to be more than 9 because instead of 16 flies, I've got 20. So it's got to be more than 9. And I've worked it out 11.25, which sounds about right. For the 2 in the middle, the 3s, I got 3.75, which again sounds about right, if we could have points of Drosophila. For the last one, the homozygous recessive, there should only be 1 in 16, and in a 20 we would expect 1.25 if it followed the same rules. Okay, so these are my expected numbers out of the 20 flies, and these are my observed numbers under the 20 flies. Okay, now the next bit on your sheet, if you're following me with the sheet or if you're following me with the chi-squared equation, the next bit's quite tough. Okay, but you do just have to follow it along. I would say this is harder to get your head round. When you're following it through, you need your observed numbers, these, I'll just put O so you remember, and these are your expected numbers, E. Okay, so when you're doing your table, you need the observed, then the expected, then the third column you do observed minus expected in each of the 4 phenotypes. Then you do observed minus expected squared, and then you do observed minus expected squared over expected. It's a mouthful. Okay, so once you've got to the end of those equations, normal equations for chi-squared, you then have to add them up. So you add up the last column for the 4 phenotypes, and you should get quite a small number. Okay, usually under 1, up to 2 maybe. I would say, looking at these numbers, so 11.25 I expected, I got 11. 3.75 I expected, I got 3, and then 4 for the other category. 1.25 I expected, I got 2. So I would say that my ratio is extremely close to a 9-3-3-1 proportion out of the 20 flies. So I would say, even just looking at it, not even looking at my chi-squared table yet, I would say that this follows Mendelian genetics, and I would say any difference I've got here, like I've got the 2 and I've got a 3 and a 4 rather than having the same number, any difference I've got is just due to chance. It's not that we have to rethink Mendelian genetics. It's not that the ratio is wrong. It's just purely chance in the random population of Drosophila I had here. Okay, so the way we find that last bit out is to tell whether it's statistically significant is because we compare to a chi-squared table. Now you can look this up on Google, it's absolutely fine, and it's just a standard kind of reference table that's used. All right, so for ours you need to work out the degrees of freedom, and it's the number of categories minus 1, and ours we've got 1, 2, 3, 4 categories, so the degrees of freedom would be 3. Okay, and we are working on a 0.05 probability. So what we need to do is 3 will be on the left side, the degrees of freedom will be on the left, and you follow it across until you get to the P equals 0.05 column, and you read off the number. Now I can tell you already it's 7.815. And my number for expected, I could work it out, but I can tell you it's a lot less than that. I don't need to work it out because I know it's going to be a lot less than that because the numbers are so close. If the numbers are massively different from this 11.25, 3.75, 1.25, I would be getting close to this 7 if the numbers were drastically different. If, say, it was 6, 5 and 7 and then none, it could be that I'm getting closer to this, but that's quite a large number to get close to with this calculation. So I would think my number is somewhere between 1 and 2, something like that. And what we say is, if you're following me with the sheet, you're looking at the last box right at the end, you're nearly done. If our value, which is the 1 point something, I'm sure, is smaller than the critical value, 7.815, we say that the difference is due to chance and is not statistically significant. And in this case, our results are so close to Mendelian ratio 9.331 that any difference we do have is due to chance. It's due to chance in the population. Mendel wasn't wrong. We don't need to rethink all the laws of inheritance that we've been following. It is statistically insignificant and it is simply due to chance. Okay, so that is your chi-squared. Very useful for inheritance ratios. Hopefully you've followed through the calculations and you've got the number at the end. And you've found a chi-squared table on Google or something similar to get you your 7.815 if you're doing this worksheet. Therefore, you need your 3 degrees of freedom and 0.05. Okay, thanks very much.